Hi, my name is Taiba Khan, and currently I'm a third year student at Bucknell, majoring in biology and minoring in philosophy. The past 2 years, I've had the opportunity to work in Dr. Morgan Benowitz Frederick's lab, where I was able to conduct this research entitled Relationship Between Aggression, Body Condition, and Parental Feeding in Chicks of the Seabird species Black-Legged Kittiwakes. The model species used in this research are black-legged kittiwakes, or Risa tridactyla, which are small coastal gulls with a large global population estimated to be over 9 million adults and colonies ranging throughout the North Atlantic, North Pacific, and Arctic Oceans. This species displays biparental care and typically broods 1 to 2 chicks per breeding season. In particular, birth order often plays a large role in the fate of kittiwake chicks. In the seabird species, hatching asynchrony occurs when a mother begins incubating her eggs before she finishes laying the entire clutch, which results in the early development of the first laid eggs. This causes the chicks from eggs that are laid afterwards to be competitively inferior and smaller than the firstborn. Size and age are relevant in species with intense sibling competition because these factors create a clear dominance hierarchy in favor of the firstborn sibling, or the A chick. The dominance hierarchy commonly results in facultative siblicide, a rare behavior where the A chick may subject the B chick to aggressive behaviors, which can result in death. In this research, we explore the aggressive behavior exhibited by the A chicks and whether there is a relationship between aggression, chick body condition, which tells you how heavy a chick is for its skeletal size, and food availability. So, in order to understand what we explored in this study, I wanted to give an overview on how we conducted our research to begin. First, we used video footage of kittiwakes from an ongoing experiment on Middleton Island in Alaska in order to make behavioral observations on the species. Each breeding season, adult kittiwake pairs nest and brood their chicks on artificial nest ledges that are attached to an old radar tower that we could see at the top here. And these nests are accessible through a one-way mirrored window inside the tower. These windows allow researchers to observe and manipulate variables such as feeding. In fact, a feeding tube is located at half of the nests, which are fed nests, and this allows researchers to feed the adults as much whole fish as they will eat 3 times a day. So essentially, the birds at these experimental fed nests aren't food limited. The remaining nests or unfed nests are not provided with supplemental food and must rely on foraging or natural means to obtain food. In the tower, researchers taped 1 hour-long videos of nests with 2 chicks when the A chick was 5 days old, and both the fed and unfed nests were recorded. So in this study, we questioned how kittiwake A chick aggression relates to food availability and their body condition. And in order to explore these relationships, we tested 3 competing hypotheses. Our first hypothesis, or H1, is that aggressive behavior is more common in stronger chicks, who grow larger because they're better able to monopolize food through their aggressive behavior. If this is true, we will see a positive correlation between aggression rates and chick body condition. The second prediction of this hypothesis is that when aggression rates of chicks in the fed nests, or the nests that are experimentally provided with more food, are compared to chicks in the unfed nests, the nests where they were not given supplemental food, we will see higher aggression rates in the fed group. In contrast, our second hypothesis, or H2, is that weaker chicks display more aggression because they need to obtain more food. If this is true, we will see a negative correlation between aggression rates and body condition, and chicks in the unfed group will display higher aggression rates than chicks in the fed group. Finally, our null hypothesis, or H3, suggests that there is no relationship between chick aggression and their body condition. If H3 is true, we will see no statistical relationship and significance between aggression rates and the variables used to determine chick body condition. Coincidingly, there will be no significant difference in aggression between the fed and unfed groups. For our experiment, we created an ethogram, which is a table that describes the behaviors or activities observed in an animal. This was important so that we could consistently identify and quantify the behaviors we saw from the video footage of the kittiwake chicks from Middleton Island. A chick aggression was categorized by pecking, squeezing, or twisting the B chick's neck, and an attack series with a pause of less than 5 seconds counted as one exhibition of aggressive behavior. We used the program Boris, or Behavioral Observation Research Interactive Software, to conduct behavioral analysis on 29 5-day-old A chicks. Using Boris, we were able to input our ethogram into the software and record timestamps for the timing and duration of each focal behavior. In total, 29 hours of video footage were observed to collect data on chick aggression in both the fed and unfed nest, and chick aggression was calculated by dividing the number of times aggression occurred by the total video time. Here is just an example of an A chick showing aggression towards the B chick. Since many chicks did not show aggression, this resulted in many zero values for aggression in our dataset. Therefore, the data was not normally distributed. In turn, we conducted a Wilcoxon test, which is a non-parametric test to compare aggression rates between the fed and unfed chicks. Our results from this test, though non-significant at a p-value of 0.1583, suggested that fed chicks tended to be more aggressive than the unfed chicks. However, these results seem to be driven by the outlier in the fed group, which had an extremely high aggression rate. Nonetheless, it's interesting to examine what these results suggested. This figure shows us the proportion of fed versus unfed groups that showed aggression at all during the hour of recording. Many chicks did not show any aggression, which resulted in zero values in our data for aggressive behavior. Thus, we ran a chi-squared analysis to determine whether the proportion of chicks that showed aggression differs between the fed and unfed groups. There was a non-significant trend with a p-value of 0.07 for the fed group to have a larger proportion of chicks that were aggressive compared to the unfed group. This result seemed to be consistent with our first hypothesis that stronger chicks are more aggressive. So here's a graph comparing body condition index versus aggression rate in both treatment groups, just to visualize the data since the distribution of data prevents normal statistical analysis. From this graph, it is evident that there is a positive relationship between body condition and aggression rate in the unfed group. When we look at the trend for the fed group, there is an inverse relationship between body condition and aggression rate. In other words, as body condition improves in the fed chicks, the chicks demonstrate less aggression. However, it's important to note that there is an extreme outlier in the fed group, which can be seen at the top left of this graph. And this seems to be driving the trend that we see here. This graph also shows body condition index compared to aggression rate in fed and unfed chicks, but instead the outlier in the fed group has been removed, so we could assess how that affects the relationship between these variables for the fed group. Removing the outlier eliminates the relationship between aggression and body condition in chicks from the fed nests. Overall, our data suggests that feeding might undo the relationship between body condition and aggression rates, as we can see a discrepancy between the relationship among fed and unfed chicks. So in general, our results seem to support our first hypothesis that stronger chicks display more aggressive behavior because the fed group tended to display more aggression and there was a positive relationship between aggression and body condition, though only in unfed chicks. A previous study conducted by Dr. Scott Hatch, Sarah LeClaire, and others in 2010 studied similar variables. They questioned how food availability affects kittiwake chick aggression, begging rate, growth, and survival, and conducted their research at the same site at Middleton Island, and they used the same feeding groups as well. Their results demonstrated that fed A chicks were less aggressive than unfed chicks, which contrasts with our results that indicate that the fed group displayed more aggression. Also, we found a positive correlation between aggression and body condition in unfed chicks, while they found a negative correlation between A chick growth rate and aggression frequency in the unfed group. Although their study compared aggression frequency and growth rate, which is not equivalent to the body condition index used in our research, these are generally contrasting results. We are not sure why there is a discrepancy between these results, but it's possible that the ecological conditions were different in the year in which the two studies were conducted, and this might play a role in the relationship between feeding and aggression. Finally, I would like to give a special thanks to a few people, including my mentor, Dr. Morgan Benowitz Fredericks, who introduced me to the study of kittiwake behavior, guided me throughout this research, taught me various forms of analysis, and helped me improve my work through several revisions. Also, I would like to acknowledge Sierra Pete, who conducted all the fieldwork for this research, worked closely with me on the behavioral analyses, and studied the trends and data collected for this project. Thank you to Chase Hohen as well, who um, helped create visualizations of the data. And I also would like to mention the rest of my research lab group, including Paige Kane, Stephanie Lynn, Verona Hendricks, and Aideen Kelly. Last but not least, I would also like to acknowledge the researchers at Middleton Island, such as Dr. Scott Hatch, who is the head of the Institute for Seabird Research and Conservation, and others who have helped feed the kittiwakes. Thank you for listening to my presentation, and I hope you enjoyed it.